Cancer is one of the hardest medical challenges out there. It's caused by a breakdown at the genetic and cellular level, where the cell's ability to regulate its own replication is disrupted, and the cell replicates uncontrollably. One of the many reasons why cancer is so challenging to treat is because there are many different types and manifestations of cancer, each with its own cell dynamics, tissue dynamics, and responses to treatment. Even worse, many cancer treatments, like chemotherapy, are indiscriminate. They harm both healthy and cancerous tissue. But the hope is that more cancer cells are hurt than healthy cells. And this can make chemo a grueling process, leaving the patient emaciated from nausea and chronic pain and exhausted. And in many cases, chemo doesn't even stop the cancer, and the patient's last weeks or months or years are spent in a state of discomfort and indignity. It's tragic, and it adds an emotional cost onto the technical difficulties of treating cancer at a molecular level. On the bright side, there are some common themes that are seen among many or even most cancers, and these common themes have become targets for potential cancer treatments. For example, consider the protein p53. This is a small protein present in all of our body's cells. It has a tumor suppression function, meaning it keeps your cells in line and it prevents them from becoming cancerous. More than half of all human cancers, and hundreds of millions of cancer-caused deaths, are associated with mutations to the p53 gene. These mutations can cause the gene to change shape, revealing the hydrophobic inner core of the protein to the aqueous solution. Now, the hydrophobic regions of all of these altered p53 molecules are attracted to each other, and so they'll cluster together in big aggregations to minimize contact with the watery cytosol of the cell. These clumps of p53 are not able to perform their tumor suppression functions, and the cell falls into high risk of becoming cancerous. Normally, all of the p53 in a given cell exists in an equilibrium between three different states. You'll have some of them in a properly folded state that are fully functional. You'll have some that are in a partially unfolded state and are partially functional. And then you'll have some that are in an aggregating state, where they are grouping together and they're not really functional. This is an equilibrium, so you have individual molecules of p53 moving between all of these states at a given time. And so long as the equilibrium is stable, the active, properly folded p53 should be sufficient to perform the cancer suppression function that the cell needs. However, mutations to the core hydrophobic region can destabilize this equilibrium by keeping more p53 in the aggregating state, and this can lead to impaired tumor suppression and the formation of cancer. Now, this is where things get really cool. Some scientists in Abu Dhabi were researching the medical value of molecular tools called protein mimetics. These are small artificial or modified proteins that are designed to replicate or mimic the actions of specific natural proteins. Now, many of these protein mimetics were developed to treat insulin resistance of type 2 diabetes, or the plaques and neurofibrillary tangles associated with Alzheimer's disease. And these worked by breaking up the aggregated clumps of proteins, allowing the individual molecules to behave normally and eliminating the harmful effects of having these large persistent aggregations. After scanning some mimetics databases, the researchers were able to identify a protein mimetic that can break up the aggregated clumps of a particular type of mutant p53 protein. This mimetic, known as tripyridylumide or ADH6, was then tested in a wide range of tissue and cancer cell types, including with p53 proteins with a variety of mutations, so the researchers could observe and measure any effect it had on the p53 aggregates and the cancer's growth. Excitingly, they found that ADH6 was really quite effective at breaking up these p53 aggregates, which allowed the p53 proteins to behave normally again, reestablishing that equilibrium and recovering the cell's ability to suppress cancer formation. In most human cancer cell cultures that were tested, this led to the tumor growth coming to a sudden stall, with the tumors losing viability, shrinking, and dissolving. Even better, as the ADH6 just targets p53 aggregates and it returns p53 to normal functioning, ADH6 doesn't have this indiscriminate toxic effect. it only affects cancer cells with mutated, altered functioning p53. It doesn't have any effect on healthy tissue near the cancerous cells. And that's amazing, because it means this is potentially a cancer treatment that won't have the horrible side effects of collateral casualties among the healthy somatic cells. Of course, this drug will need to be run through some clinical trials first, and there's a lot more research that has to be done. But these preliminary data are mind-blowing. This drug appears to have almost everything we want in a theoretical ideal cancer drug. Let's list them off, and you tell me if I'm missing anything. It targets a specific cellular structure, so we know where to look to study the mechanism of action and to make improvements. It doesn't introduce new byproducts or kill the cell directly, but instead it simply breaks up a problematic structure and lets the pieces, which are natural parts of the cell, return to their normal, natural state of functioning. This natural state of functioning is a perfectly healthy in-house method of cancer control, which leads to the self-destruction of the tumors. And nearby healthy tissue is never at risk, avoiding all of the complications and terrible symptoms of treatments like chemo. Let's hope this drug makes it through trials and gets into your local pharmacy, because, from where I'm sitting, this looks really incredible. We're seriously looking at a drug that, potentially, could save many millions of people from suffering one of the most common kinds of death that humans experience: death from cancer.